Next slide. So why is this important? Hepatitis B is a major cause of morbidity and mortality around the world. This is recent data from the Global Burden of Disease study that I found quite interesting when looking at causes of disability-adjusted life years lost from primary liver cancer among young people. Hepatitis B is far and away the leading cause of this morbidity and mortality related to liver cancer, around the world, far outpacing, other causes. That can change with access to current treatments that are available that can reduce with one pill a day that are safe, very affordable, and can reduce the risk of liver cancer by 40 to 50%. However, very, very few people are diagnosed with hepatitis B, about 10% estimated globally, and only about 2% of people receiving these therapies, based on previous, recommendations. So that's the environment that we are seeking to change to make these treatments that we have now more readily available as we look forward to improved therapies that are currently in development. Next slide. To start off with, screening is important. And I just wanted to make note that WHO has previously developed recommendations for hepatitis B screening that were released in 2017, pointing out different strategies that could either be lab-based or point-of-care based for HB surface antigen testing to detect people who are infected with hepatitis B virus, and then that being followed by nucleic acid testing that could help you make treatment decisions and monitor response to treatment. Next slide, please. And so the guidelines that were prepared by WHO were released in March of last year, and these are the links to those, and I encourage you all to go through those documents. They're very comprehensive in laying out the evidence base and the rigor of looking at the evidence that formed the guidelines that we'll be talking about today. Next slide. Again, as it relates to testing, there was a recommendation to follow a positive surface antigen test by a measure of viral load that can either be done within the laboratory or various point-of-care DNA assays strategies, and then that to be followed by reflex. There was an encouragement to have a reflex approach whereby the same specimen was used for both surface antigen and nucleic acid testing. And then the recommendations went further to encourage reflex delta testing for persons that were infected with hepatitis B. Next slide. So this is sort of the summary that we'll be going into in more detail in the coming presentations. But basically, the recommendations from WHO presented different options from which to determine eligibility and benefit of treatment for persons that were HBsAg positive, based on their with evidence of fibrosis, based on serologic testing for APRI score determination, transient elastography to look for evidence of fibrosis, or a clinical diagnosis of severe fibrosis or cirrhosis. Alternatively, one could use HBV DNA testing alone and determine to identify persons that had a viral load greater than 2,000, coupled with evidence of liver inflammation, as shown here. There was also a recommendation, or you could look at presence of co-infections, various comorbidities such as adult onset diabetes, steatotic liver disease, immunosuppression, or extrahepatic manifestations of HBV infection, or in the absence of access to HBV DNA assays, just rely on abnormal ALT levels alone for someone who was HBsAg positive. So we'll explore these in a bit more detail coming up. Next slide, please. I think the other interesting aspect of the recommendations is really focusing on testing and antiviral therapies for pregnant women that are HBsAg positive. Over and above the current recommendation for antiviral prophylaxis for pregnant women with a viral load greater than 200,000 international units, there was also a conditional recommendation of placing all HBsAg positive women on antiviral prophylaxis. So we'll again be exploring that here in a moment. Next slide. And then lastly, now that we in particular have a therapeutic option for Delta infection, increases the value of testing for Delta. And there was a recommendation, as I mentioned earlier, for reflex testing for persons that were HBsAg positive to proceed with Delta testing to identify co-infection with HDV. Next slide. Now, the real benefit here is that this is going to expand the eligibility of the persons that are HBsAg positive for HBV treatment from 19 to 25% based on a meta-analysis looking at the impact of the previous recommendations to at least a 50% of persons being deemed eligible that are HBsAg positive with these new guidelines. So the guidelines have expanded the number of persons that are now eligible to be treated for hepatitis B. And secondly, next slide, please. The other element that we'll be getting into is that there, in addition to expanding treatment eligibility, the guidelines are seeking to simplify treatment by using these different channels and then recommending ways of integrating hepatitis B into other care services, simplifying the guidelines so that they're more easily understood by non-liver specialists, and recommending different strategies to apply these guidelines throughout health systems. And so that's what we'll be exploring also in our coming presentations.